All right, looks like we are live, and I want to welcome everybody to Standing for Truth. My name is Donnie, and I am your host for this evening's important presentation. We are going to be answering the question, why did God create viruses, bacteria, and other pathogens? And I am thrilled for a couple reasons. Firstly, I'm thrilled to have Dr. Jerry Bergman back with me to speak on this important topic. And I've been sick for about a week, and so it is great to be back in the studio here and bringing you some awesome content. Now, I do want to recommend people check out this book right here. And so this is Dr. Bergman's newest book, titled the same title that we've given this presentation. Why did God create viruses, bacteria, and other pathogens? And so this is a must read. Excuse me. And so please do check the description box of this video to pick up a copy. All right, before I hand it over to Jerry for his presentation, I do want to give him a proper introduction. And so Dr. Jerry Bergman has taught biology, genetics, chemistry, biochemistry, anthropology, geology, and microbiology for over 40 years at several colleges and universities, including Bowling Green State University, Medical College of Ohio, where he was a research associate in experimental pathology, and the University of Toledo. He is a graduate of the Medical College of Ohio, Wayne State University in Detroit, University of Toledo, and Bowling Green State University. He has over 1,300 publications in 12 languages and 40 books and monographs. His books and textbooks that include chapters that he authored are in over 1,800 college libraries in 27 countries. So far, over 80,000 copies of the 60 books and monographs that he has authored or co-authored are in print. Jerry's also appeared on the Standing for Truth program somewhere between 8 to 10 times. He's a frequent guest, and he always gives an excellent presentation. And so, Jerry, I appreciate your time that you've given to us tonight. I'll hand it over to you. How you doing tonight, Jerry? And then we'll get into your presentation.
I'm doing pretty good once we got the computer system to work.
Right.
Which is always a challenge. And this is a topic which I think is important, and many people have told me that it is very important. Why did God create germs? Why did God...
Jerry, before you continue, if you don't mind, sure. If you see at the bottom there, it should say hide. And that way it won't.
Okay.
Yeah, can you use, yeah, right there.
Okay, now I don't see my picture.
It did hide the, now you just have to make sure to go back to your PowerPoint, wherever your PowerPoint slide might be. I think I can see it behind the, so if you minimize the StreamYard.
Okay.
Yeah, there it is. And then just make sure to click the actual StreamYard box or window. Yeah.
Okay, and then now can I click hide again? Is that?
Yeah, click that hide. Left click it.
Okay. Okay. Now we're good. A good example is a man by the name of Charles Templeton, who was a evangelist and very popular, in fact, more popular than Billy Graham. And let's see if I can get this to work now. Okay, there we go. And you can see he is here doing a seminar. I think he's from Toronto. And there you can see Billy Graham doing the same thing. And he not only left the faith, but he also became an atheist. And why he talks about in his book, Farewell to God, My Reasons for Rejecting the Christian Faith. And one of the reasons he gave was, well, evolution was one. How can one believe in the biblical account of creation when every eminent scientist agrees that all living species have evolved over millions of years from primitive beginnings? So that's one reason, which we won't cover tonight. But he also said God's grand design also includes the creation of parasites, worms, fleas, ticks, lice, bacteria, viruses, and the aberrant cells that attack the body, the organs, or brain. In other words, cancer cells. and the malignant cells that stimulate the development of diseased tissue and causes death and suffering. So why would God create, especially the focus now is on germs? Especially since Genesis said, and God saw everything was very good. The Genesis fall caused corruption that affected the entire creation. So everything originally was very good, but the fall occurred. Thus, we will expect an imperfect world, which is what we see around us. And this is the original cover I had for my book. And my publisher said, no way. And it shows death, although this is not a girl, this is a clay model. In fact, it's done by a very famous artist in Germany, and he is trying to show disease, and it shows it too well. So several people said, oh, got to change the cover. It won't work. Okay, where are bacteria located? It may surprise you that the human body, has trillions of bacteria. Majority are in the gastrointestinal tract, and all surfaces on contact with the outside world, like the skin and the respiratory tract and the genital tract. These all are covered with, bacteria. And the rest of the body is sterile. It should not have any bacteria. When it does, then the immune system is triggered. And it may surprise one to know that 99.999% of all bacteria are good. In fact, harmful bacteria are very rare. And you need good bacteria to be healthy. And it may surprise some people that a newborn is sterile, although not for long. Well, how do they get bacteria in their gut? Well, one way is that babies are just very cute. They're very kissable. And so when you have a newborn baby, you kiss the baby, and the baby then picks up from your kissing bacteria. And the bacteria then takes a journey, goes in the mouth, goes in the gut, and populates, I should say, the bacteria in the entire intestinal system. And that bacteria has to be there for a couple of reasons, which I'll talk about in a minute. As a matter of fact, too clean is a problem. A good example is we had a patient at the hospital that she got the idea that we need to protect our children from germs. So she protected her child from germs to the extent that she sterilized the sheets, the clothes, and so on. And so he lived in not a totally sterile environment, but as far as she could get a sterile environment. And the problem with that is that for the immune system to develop properly, it must be exposed to bacteria, so it learns to differentiate the good guys and the bad guys. And unless it is exposed to bacteria when you're young, it cannot do that. And a good example is allergies. Allergies is where the body encounters something strange, and therefore it responds by thinking it's a problem. But if you're exposed to things like peanuts when you're young, your body will realize that, no problem. And therefore, when you're exposed to peanuts when you're older, you will not have a problem in most cases. And so early exposure is critical, not only of bacteria, but also of peanuts. In this case, that's a well-known allergy, and that's why I use that example. Okay, what causes bacteria to become pathogenic? The main cause is mutations. And almost all the bacteria that are a problem have mutations, which causes them to be a problem because something's changed. They're not the way they normally are. There's a change which causes a problem. Plasmid transfer is another means. Plasmids are little DNA circles, and they carry information on them, and bacteria will transfer these plasmids to other bacteria. And in case the plasmid has a mutation, has a problem, and if that is transferred to other bacteria, that can spread the pathogenicity to other bacteria. Another major problem is weak immune system. If you're exposed to pathogenic bacteria, many, if not most, not a problem if you have a strong immune system. And at age 50, the immune system is about half as strong as it was at age 20. So as we get older, the immune system weakens, And as a result, we do not have the defense that we need. This is why when you're young, primarily, no problem. The exposure is not a problem because your immune system can effectively take care of the issue easily without any feedback. That's a problem. Also, if the immune system is compromised, like you have had AIDS, therefore the T cells are damaged, and therefore that is going to make you susceptible to bacteria, which you normally would not be. Another problem is it invades a non-host organism. There are a lot of organisms, the best example is HIV, which in its host organism, no problem. All pathogens, by the way, have a host organism. For the HIV and the monkeys, no problem. They get along very well. When that bacteria ends up, or virus in this case, ends up leaving the host organism and jumping into another organism like humans, then we have a problem. Another issue is Staphylococcus aureus, which is the skin bacteria. All of you right now have Staphylococcus aureus on your skin. And when you have a cut, then the staph infection can get into the body and cause problems. So where it is on the outside of the body, no problem. When it gets into the body, then it can cause a problem. In this case, it can enter the bloodstream, even the joints, the bones, the lung or heart or other organs. So when you have the bacteria, which is normal, it should be there, but when it gets into the wrong part of the body, that can be a problem. Now, normally, again, your body will take care of this, but not always. Probably the best example is E. coli, Escherichia coli, O15787. This is a mutant form. And this, by the way, can be lethal, especially if your immune system is compromised within a few days or months. Now, if they know you have this O15787, then they will, of course, administer medication, antibacterial agents, which will help deal with the problem. And again, E. coli is in your gut. You have zillions of them. In fact, fecal matter, the dry weight of fecal matter is about 40% bacteria. So there's a huge amount of bacteria which is excreted every day. Some mutant strains, though, can become pathogenic, and they can become pathogenic by the plasmid transferring from one to another, as I mentioned. And so that is an issue. Also, transposons, bacteriophages, and so on are part of the story. This is a picture of Staphylococcus aureus, the skin bacteria. It's a spherical, circular bacteria. This example is stained. You normally can't see it, so they stain it to help one see it. But you can see they're just little ball-shaped things. And the problem is from home to another organ. And the best example is the appendix. The appendix has a number of bacteria, usually good bacteria, and the appendix is a safe house for bacteria. So when you take antibiotics and it destroys the good bacteria, you need to repopulate that area as soon as possible. And one way of doing that is the appendix, which releases that bacteria into the peritoneum and other areas which are, or the peritoneum would be a problem. When you release it into the gut, then it can help replace that. When you have antibiotics, the doctor should tell you to consume some antibacteria issue like yogurt's fine. And so you have a probiotic which replaces the bacteria which is lost in the gut. Okay, moving on. When I took microbiology, by the way, many years ago at Wayne State University, he said in one of his classes, bacteria are our friends. We need to realize that they are with us for good reasons, and we need to make sure that they are healthy. And of course, I was 20, I guess, 19, and I thought, what a dumb statement. But he, in the class, helped us understand how indeed that was very true. Okay, sources of probiotics. And these are things which facilitate the growth of bacteria, or which actually have live bacteria, which will populate the gut or wherever. Yogurt, the most common example, most well-known example. Sauerkraut, another example. Apple cider vinegar, another example. Apples, olives, pickles, garlic, and there are many other probiotic sources. So just general health, they recommend that you include these foods in your diet. Okay, what do bacteria, probiotic bacteria, what do they do? Well, they control candida yeast infections. They help manage digestive health. They clear the skin. So if you have a good kind of bacteria on your skin, it protects your skin from the problematic bacteria. Reduces food poisoning, and it's important for the immune system health, controls nail fungus, and it kills other pathogens which are a problem. Helping degrade the food, does that in many ways. It can neutralize toxins, which is important because in all foods you have certain levels of toxins, and so they must be neutralized so they don't cause a problem. Protects the body surface, the skin, the gut, and so on from pathogenic bacteria. One of the more important roles it has is to manufacture vitamin K, thiamine, folate, biotin, riboflavin, cobalamin, niacin, pantothenate, pyridoxine, and pantothenic acid are all manufactured by the gut bacteria. So if you don't have gut bacteria, some of these, especially vitamin K, will not be manufactured as it should. They also are important for carbohydrate metabolism, so they help digest your food. They have a role in protein and lipid homeostasis to make sure that we don't have too much or too little, to ensure that we have the right amount of proteins and lipids of certain types. And importantly, the enzymes they make are required for B12 synthesis. And therefore, the only way we can get B12, aside from meat, non-vegetarian sources, is from the bacteria. And it helps maintain reproductive health, vitamin B12 does. And unfortunately, we focus in the past on destroying bad microbes. And it may be more important, in fact, it is more important as a whole, that we take good care of the ones that are good, because they will fill up the apartments so that the bad bacteria will not have a place to stay. Here's a good photograph of bacteria. This is the worm-like, it looks like a worm, it looks like a hot dog, I guess, a type of bacteria. And the names of bacteria are based partially on their shape, and you can see the shape here. Again, these are colorized, but it gives you a good idea of what they look like. Okay, how do they serve humanity? Well, they manufacture ethanol and enzymes. So when ethanol is produced in factories, well, beer, of course, wine is the best example. To produce quality, good beer, you have to have the right bacteria. And if you don't, you're going to end up with some poor wine. In fact, they have to check constantly the bacteria to make sure the bacteria producing the wine is of the type, or bacteria producing the beer is of the type necessary to produce the flavor that they want. They make drugs, antibiotics, and vaccines. They make biogas or methane. So the methane we use called natural gas, is made by bacteria. Cleans up oil spills and toxic waste, kills plant pests, and it's critical for gene therapy. That's one role that I had when I was at the medical school. We used gene therapy, and we used lambda, usually virus, which transfers very effectively normal genes to human cells. So if the genes are defective for some reason, we can transfer normal genes in certain cases to the cells. Now, a good example of pathogens is from John Snow, a medical doctor. And in London, there was an epidemic of cholera, and hundreds of people were dying, horrible disease. It doesn't take 4 or 5 days and you're dead. It's a big problem. And the concern was, how did this epidemic occur? What was causing it? And so John Snow, what he did is he charted all the cases in the city of cholera. And he found most of the cases were right around this pump, the Broad Street pump, which is where you pumped and got water from. And the ones who were not living nearby that pump, we know now were drinking water from that pump because they visited somebody or they had some contact with that pump. So Dr. Snow said, you want to solve the problem? You put a chain on the Broad Street pump and the problem will end. And of course, they thought he was nuts. But, they had no other solution. So they finally turned it off, the pump, and guess what? The cholera epidemic abated until there was not a problem. Now we know that the problem was there was a sewer line nearby. The sewer line was broken. It was leaking water and sewer fecal matter into the water that people were drinking at the Broad Street pump. And so now that helps us understand a major spread of bacteria is from fecal matter. And it's interesting that Deuteronomy 23:13 said, and I'll just summarize what the scripture says here. Carry a shovel with you whenever you travel. If you're going somewhere, carry a shovel with you. And when you've relieved yourself, use it to dig a hole, then refill it, covering your excrement. Basically, the rule was, Josephus in his book on the Jewish history, basically the hole was supposed to be a foot deep, and then bury fecal matter and cover it up. Well, that's pretty clear why. We know now why. And they probably knew why back then, but this was ignored for years, and no wonder we had problems. So now we have very strict rules relative to fecal matter, and therefore we have far fewer problems with the spread of pathogens. So part of it is the pathogens are there, but we need to take care of them properly, which by and large most cities do. Quarantine is another important scriptural rule. There are 16 verses that talk about quarantine, and basically when one is sick, you are to be quarantined for, in this case, 7 days, but this is a disease, leprosy that was common at that time. So basically, they quarantine you until you have no signs of the disease, and then you can go among other people. So quarantine was a very important way of reducing the spread of disease. Now, talk about leeches, and this is probably one of the ugliest faces that I've ever seen. That's the face of a leech, and they are used for therapy, which may surprise people. So why are they used for therapy? Well, that's a profile, which they're not quite as ugly, but still not exactly cute. But you can see that they're sucking blood. The front mouth part is hooked up to the arm here, or the leg. I think it's the leg, and then sucking blood. So what we know is they attach themselves to the body. They live off blood. Number one, they improve blood circulation. Very important. They also release a powerful blood thinner, and so clotting is less of a problem. They also release compounds to reduce the pain. And so they suck the blood, improving circulation. And after a skin graft especially, the main issue is circulation. So the leech will improve the circulation in that area. It will then reduce the likelihood of blood clots, and by producing the blood thinner, and it produces a strong anticoagulant, herulin, and it also treats other problems such as the pain. It reduces the pain, and the leech can also be used for other body ailments, including inflammation, skin diseases, even rheumatic pain, even reduce certain reproductive problems. Maggots, another ugly creature, it may seem. These are fly maggots. There they are. What do? Well, military surgeons noticed soldiers with maggot-infested wounds were healed much better than the infestation-free wounds that the soldiers had. And so they knew back then, this is before the Civil War, they knew that these maggots, for some reason, had a beneficial effect. And then it wasn't long, and we began to use them to treat medical conditions. And now we know why. Probably one of the best examples is when you have a diabetic foot infection, poor circulation, and the doctor, when he tries to save the foot, scrapes away not only the good tissue and the bad tissue, but also tissue that shouldn't be scraped away. They can't differentiate very effectively, of course, the good tissue from the bad tissue. And now we know that when you have diabetic wounds, one of the primary alternatives, aside from losing your leg, is using maggots, which effectively differentiate necrotic tissue from good tissue. So they selectively remove the bad tissue and leave there the good tissue. So they're incredibly effective at separating the problem from the body. And they also heal wounds by ingesting not only necrotic tissue, but also by reducing bacterial infection, which is a big problem. And we use it not only to treat wounds from injury, from diabetic conditions, but also burn and accident victims. It's used, even treats osteomyelitis, which is a bone infection. And so therefore, they can be very, very useful as well. In fact, the success rate is quite good. When I was speaking not too long ago, one of the men in the audience lost part of his leg. And when I asked him about this, he said, well, I was diabetic and the infection just spread. So to save my life, they had to cut part of the leg off. And my thought is, why not use maggot therapy? He said he never heard of that. Right next to him was a nurse who said, oh, in my hospital I work at, we've got whole jars of maggots. They're commonly used to deal with many conditions. So they're out there, but I guess they're not in every hospital, or every doctor's not aware of this type of therapy. You might think it's kind of gross, but given the choice of losing my foot or some maggots wrapped up in doing its job, without thinking twice, let's put the maggots in there. Let's deal with this. It tickled a little bit, but by and large, they're not painful for a couple of reasons, actually. I guess I don't mention here, but they produce a compound which not only reduces the pain, but also reduces the irritation. And so therefore, it can be a very effective means of dealing with this problem. Mosquitoes, what about mosquitoes? Certainly they are not good. Actually, the vast majority of mosquitoes are beneficial for a number of reasons. They are good pollinators. They take care of more problematic insects. The only mosquitoes that are a problem are the females, because the females have to have blood in order to produce the nutrients necessary to allow their eggs to grow up to be normal mosquitoes. And actually, most insects are very helpful to humans, at least not harmful. In fact, there are very few really that are harmful. The female mosquitoes can be harmful if they bite somebody with malaria and then bite somebody else that doesn't have it. They can then pass the malaria pathogens from the malaria patient to germ-free patients. So that can be a problem. But on the other hand, if you're in an area where there are no issues with malaria, like most of where I live, most of Michigan and Ohio, they rarely cause malaria because they're a vector. And if we don't have any malaria patients in this area, they're not carrying anything that's a problem. Okay, what else they do besides pollinating? They disperse seeds, they recycle dead plants, animals, and dung. They also can control plant and animal populations and provide subsistence for other animals. There are many animals like spiders that a delicacy for them is mosquitoes, and therefore it is a food source for them. In case you're curious, the male you can see has these extensions. The arrow there shows the extensions, which are hairy. That's male, which are not the problem. The female you can see does not have these hairy extensions. They have a fine line, thin extension. And these are the animals, the mosquitoes, that are the problem. The males are also smaller than females. But again, by and large, they're only basically irritating. And by the way, they can determine we're around by the carbon dioxide that we release. And so they're attracted to carbon dioxide. And so if you can put some smell thing, like some of the anti-insect protectants, they produce a smell which masks the carbon dioxide, and then mosquitoes will leave you alone. The males, what do they feed on? Well, plant juices, such as nectar. And of course, they need a lot of sugar, and therefore that's what they do. Okay, mosquitoes are also important, which surprises me when I was doing research for this book, because I haven't read a great deal about mosquitoes. It's not one of my main interests, but I found they help control rodents, especially rats, mice, and rabbits, and play a significant role in many aquatic food chains. In other words, they are a meal for many, many animals. And they bridge the gap between the fish and the near microscopic organisms. So they're kind of in between size-wise. Spiders, most people know that spiders were beneficial, especially farmers. The reason they're beneficial is because their webs catch just about any insect that could be out there. And so they reduce the problematic insect population. And so most people who have spiders in the barn or even in garages just leave them alone because they can reduce the problems. Okay, we read in the Bible that God said all creation is very good. Some people, especially after the COVID epidemic, say, wait a minute, not viruses. Viruses are not good. Well, surprise. Charles Darwin said, why did God make pathogens? And well, actually, God didn't make pathogens. Well, except viruses. Let's look more carefully at viruses. Here's a National Geographic special, The Mystery of a Virus. And when I read this, I thought, I couldn't believe it. National Geographic is actually ahead of the game. It says at the bottom here, they kill us by the millions, but without them, life is impossible. When I do presentations in churches, I suggest that they make sure, kind of tongue in cheek, but they should make sure that they thank God for viruses, because without them, you would not be alive. What do? They do lots of things. The interesting thing is that they rarely cause problems, and therefore, why study them? When you're raising money to study a disease, well, people will donate, for example, for breast cancer or lung cancer, because obviously that's a problem. But when you collect money to do research on viruses, people will say, well, what problem do they cause? Well, none. Well, why do you want to research them? Oh, just curious. Well, I'll give my money to some research that can help cure disease. I don't want to give my money to satisfy your curiosity. That's not what I want to do with my money. Well, so we are slowly learning what viruses do, and we don't know what many of them do, if not most of them, because they don't cause a problem. In fact, you have 5 viruses around your eyes, and we're not sure what they do. I think what they do is reduce the likelihood of bacteria and inflammation and problems. But we don't know for sure what they do. We know they're there, and we know when they're not there, which is rare, but occasionally they're not there, we know that they cause problems. Again, we have the reservoir problem. If the virus is in its reservoir, its host organism, no problem. When it gets into you, like monkeys, the HIV, from monkeys to human, a problem. The flu, same thing. Swine or ducks, no problem. When it gets into humans, it can be a problem. And viral diseases are many: polio, hemorrhagic fever, diarrheal disease, smallpox, flu, HIV, Ebola, hepatitis A, E, etc. Not many good things, these type of viruses, but again, they're rare. The vast majority of viruses are helpful. And there you can see smallpox. I think we're more aware of virus killers because they cause epidemics, and also they produce horrible effects like the smallpox, you can see. And when we look at the viruses, we can see they're very complex. In fact, we can see they're designed specifically for the role that they have. And the picture, the blue is cells, and then the little yellow particles is the HIV. Okay, what do they do? What are some of the good things they do? A major thing they do is they keep bacteria in check. Bacteria double every 10 to 20 minutes. If you take 2 bacteria, and if they could double for a couple of days, they don't run out of food, they're not killed, they would cover the earth with bacteria. They're incredible. Well, 10 to 20 minutes, they double. 2, 4, 8, 16, 32, 64. After a day, you end up with a huge number. In fact, they soon would consume all the organic material on the earth. And they would grow to 10 to the 110th power. Of course, they can't do that, but if allowed to, they would be able to. Viruses are actually not living. They have a protein shell. They're covered with markers for identification. They're ID tags. They call that an antigen. And inside the virus, we have a genetic code. We have two types, single RNA or double-stranded RNA or DNA. RNA would be retrovirus. And they have some protein. Not much there, but indeed they're very ingeniously designed for what they do. And one thing which surprises people is HIV cannot get into your cells unless it has two keys. It must have a CD4 receptor and a CCD receptor. They must have two receptors, two keys to get into your body. They can't get in if they have one. They've got to have both keys, and the keys are designed in such a way to where once they activate the lock, they can get into your body. And okay, here we go. CD4 receptor. And they must also have the key for the CCR5, which is called a co-receptor. And that's the only way the HIV can get in. So if you have a mutation in the co-receptor, you are not going to be infected by HIV. And there you can see both receptors are activated. And this is one of the most fascinating examples of viruses. And this right here, you can see is a bacteriophage. This happens to be T4. And it looks like a rocket ship. And that has particular significance for the intelligent design movement. There is a man by the name of Michael Behe who was doing research. When he looked at that virus, the bacteriophage, he realized there's no way that could evolve. It's got to have every part before it can infect and basically draw nutrients from its source of food. So it cannot survive unless it has all of these parts. And natural selection, how's it going to survive until it gets all these parts? And when you look at it, you can see the picture right here. It's not the best picture, but hey, you've got to magnify these, I don't know, several million times to see it. But it looks like a rocket ship. It looks like it was made. And he saw that. He said there must be an intelligent designer. And Behe was an important founder of the intelligent design movement, which now claims hundreds of thousands of followers throughout the world. So it revolutionized, in this case, not only medicine, but also the origin of life and evolution by Michael Behe's discovery. And bacteriophages have a very important function. They, as you can see here, better pictures of it. You can see here the parts. And you can see we even have the molecules necessary in order to produce the whole thing. And there's the DNA inside, which they inject inside of the bacteria. And there you can see actually a electron microscope picture of one. And there you can see they're attacking this bacterial cell. So this is a bacterial cell, and these are the bacteriophages. And they are very, very important in dealing with bacterial infections. And in fact, they're so important that some countries, even today, instead of using penicillin or antibiotics, they use bacteriophages to treat disease. If you're allergic to penicillin and other antibiotics, one solution is, well, bacteriophages. Put these in your body and they will take care of the bacteria that causes the problem. And why this isn't practiced more commonly, I think the main reason is we just are sold to penicillin and it's available everywhere, and it's easier just to give someone a shot of penicillin than attempt to give them the bacteriophage. Anyways, they attack only their host bacteria. So they're specific in the bacteria they attack. They do not attack human cells, and they're very good candidates to treat bacterial diseases. You don't have to worry about developing immunity or developing bacterial strains that are immune to antibiotics. As far as we know, there's no concern in that area. And it's been abandoned except, Russia, Georgia, it's a country in Asia, and Poland. still use the bacteriophage as a means of treating bacterial infections. It's called the phage approach. And what else do bacteria do? Well, they're everywhere. 1 times 10 to the 31, which is a big number, estimate on the Earth. The estimate is 1 times 10 to the 80th is the total number of electrons in the entire universe. And here are the number of stars I use here. Found everywhere, hot environments, cold environments, salty, non-salty, inside the Earth, everywhere you find viruses. Even in Egyptian tombs, they seem to last, well, they're 2,000, 3,000 years old. They open up the tomb and they find these viruses are viable. Well, they can't die. They don't die. They can be destroyed, damaged. But if they're viable, they can be removed and cause infections. In your body now are trillions of viruses, and all doing their job without nary a complaint. So they're working hard for whatever they do. They're critical for research. We use lambda virus in my work, which is a big virus. It's got plenty of room. We can pull out lots of genes and put lots of stuff in there. And therefore, they're very good to deliver as a vector certain genes to certain parts of the body. And controls harmful or damaging organisms in various areas, agriculture and medicine. They're used in a lot of cases we're really not aware of because they're used so successfully. We just, unless you're in that field, you're not going to become aware of what they do. So they're the basis of gene therapy. And the bacteriophages, mention them. They keep bacteria numbers in check without harming the human host. So it's an advantage there. Virus use in mammals. Norovirus, important for mouse intestine immune system, can actually replace the beneficial effects of certain gut bacteria, which are decimated by antibiotics. And of course, we have immune system protection. Gastrointestinal tracts of mammals are plush with viruses. Again, we don't know much about them. We know they're there. We know they have a high number. And as time goes by, the virologists now realize that we've got to basically let people know that they're our friends. And we need to understand what they do because they can help cure diseases. Other uses, they're important for wasps. Wasps produce and use viruses. And they have functionally infective virus particles. The diversity of parasitoid wasps is enormous. Helps plants survive. One of the main things that they do is they're everywhere. They use to treat bacterial infections, which I mentioned, and they can be used to kill resistant strains of bacteria. And this is important. They're part of the food chain. A variety of sea animals cannot swallow large fish. They can swallow gulps of water, and in gulps of water, in fact, the average glass of water has about 150 million viruses. So many smaller fish can get their nutrients by just floating around in the water with their mouth open, taking in the 150 million viruses without doing a thing. And these are small fish typically. And then the small fish, of course, are consumed by larger fish, and eventually we get up to very large fish, which are consumed by humans. So they're a large part of the diet of many sea animals are viruses. And again, the problem is mutated viruses. They end up in an incompatible host. Common cold, the flu, and so on are good examples. And they thrive in their proper host, no problem, but cause problems when they're in an incompatible host. And so therefore we should thank God for viruses. And as I mentioned, they serve as vectors, which lambda virus is the one that we use. That's pretty commonly used. They can be used for insecticides. They can increase the drought tolerance in some plants. And they serve as seeds to cause rain particles that allows the water to condense around them and produce rain. Coronavirus has spike proteins. And Again, we're trying to understand how they work. And the problem with the coronavirus is that normally the vaccination that we utilize has decades of research under it. With the coronavirus, they're trying to get it out within 6, 8, 10 months, and it was improperly researched, and now we're suffering some problems because of this. And there you can see the Lambda virus, which is, you can see that about every part of the virus, we know what it does. So we've got all these parts labeled so that we actually know what it does and what it needs to have to function. And that's the end of the show. That's it.
Jerry,
you got
yeah, as usual, fantastic presentation. That flew by, and you did a really good job at covering essentially everything in this book here that I want to recommend everybody check out. So I've got it in front of the camera. Why did God create viruses, bacteria, and other pathogens? And so I think this presentation is a good overall snapshot, Jerry, of the overall content and material found in your book. I like your slide near the end that the headline was, "Thank God for Viruses." And you more or less find that in the conventional literature, as in you can find papers and articles where they point out reasons why the world needs viruses, the benefits of viruses, the benefits of bacteria. They even go as far as saying you wouldn't want to live in a world without viruses and bacteria. But what they don't do, Jerry, is give the glory to God. But that's what I love about this presentation, is you give the glory to God as for why we have viruses and bacteria. And so we got a lot of questions. We got a lot of good questions from the audience, Jerry. And so why don't we just get right into it? We'll start. Okay, I guess we'll start kind of right at the top here. So question from Matt. Why did God build into our genes cancer, knowing that a mutation would cause it to manifest?
Okay, very good question. Cancer's due to breaking something. It's like we have in our car built-in brakes, stops the car. If those brakes are broken, the car will just move forward until it collides with something. Well, basically what happens in cancer is the brakes are broken. And the brakes are not only broken, but the system which makes sure the brakes are working is broken. P53 is probably well-known, called the guardian of the genome. And P53 makes sure everything is doing okay. If it's not doing okay, P53 will halt the cell division, and P53 will initiate a repair system, repair the damage. And if the damage cannot be repaired, P53 will initiate apoptosis, which basically is the self-destruction of the cell. And so this very complex mechanism works pretty well for billions of cells for your whole lifetime. Every now and then, though, especially carcinogens, break the system so that the cell is not able to stop and repair itself. P53, for example, about half of all cancers, P53 is damaged. And so therefore, you don't have this protective system. And therefore, the cell does not repair itself. It goes into the next cycle and over again. And therefore, we end up with damaged cells, which accumulates, by the way. It accumulates more and more. And after a while, it accumulates to the point where the cells are no longer function at all. And eventually the patient dies. And so cancer is a matter of something breaking the system. And there's a genetic component. If I inherit, for example, a damaged gene, that will make me susceptible, like the gene that causes breast cancer. If I female inherits that from the mother, then that female will be more likely to develop breast cancer because that gene is damaged. So the genes are the main issue, especially inherited genes that are damaged. And that's the main issue which we need to look at. And that, of course, will be caused by mutations, and mutations are a result of the fall. We know the mutational buildup has been occurring for 6,000 years. We know that 1,000 years ago, people had far fewer mutations. We know the mutation load is increasing with each generation, and therefore we have a lot of diseases that they didn't have. My guess is if you go back 4,000 or 5,000 years, you're going to find, if any cancers at all, you find they're rare and their diseases are more common. And doctors realize this. You have a medical background, so you can probably relate to this quite readily. But they realize this, and therefore we realize that there are diseases coming up. We don't have any cases yet. They're coming up called new diseases or orphan diseases, and we need to keep our eyes open. That may be a new disease that no one has ever known before, no one has ever seen before. And so the mutational load is increasing. So doctors don't have to worry about going without a job because mutational load is not going down, it's going up, causing more diseases. About 5,000 diseases now that are caused by mutations. And cancer is only one of them. And again, there's a lot you can do to reduce the likelihood of cancer. Have your genes evaluated. I had mine. My father died of pancreatic cancer. And so they tested me and they found that I had one of the genes. And so I'm checked every year. They do an MRI and they make sure that this cancer hasn't started. So now if it starts, they can hopefully nip it off before it spreads. And so they can catch it early. And so I recommend, especially if you have a certain cancer in your family, have it checked, and then they can make sure that it doesn't progress on to the point where you have the live cancer. So anyways, it's probably more than you wanted to hear, but
no, that's great.
This, so I'm pretty aware of.
I could listen to that answer on this topic for all day, basically. A lot of great information. So would you safely say then, since you pointed out how a lot of these cancers are inherited through mutations, damaged genes, then it is safe to say that the rise in cancers, the rise of a lot of these diseases, as you've said, 5,000 mutation-related diseases, is due to the increasing genetic load over time.
Oh, yeah, that's part of it. But also part of it is, the best example is smoking and tobacco. We know tobacco produces carcinogens, and we know it causes lung cancer. And interesting, my uncle died of lung cancer, smoked his whole life, 59. My uncle died of lung cancer, smoked his whole life, age 59. And so we see in the hospital where I worked, we can see so many were smokers and were exposed to other carcinogens and ended up dying young because of the exposure to the carcinogens found in smoke. Now, some people always ask, why do you have some people like the comedian George Burns, who smoked, 4 cigars a day his whole life, lived to be 99. He died of falling in the bathtub. Never had an issue with cancer. Well, the body is really very effective in repairing this damage. And his body was very, very effective in repairing his damage. He got the damage, certainly from smoking as he did. He got the damage, but his body fixed it for his whole life. And so if our body is not quite as effective, then the damage, which we all get, including George Burns, can be repaired. Or if we're not as healthy, it's not repaired, and therefore we end up with cancer.
So would you say then, because mutations cause cancer, as you're saying, but also environmental modifiers, basically like tobacco, smoking, so on and so forth, would that be considered epigenetic degeneration then, since these are external?
No, it'd be genetic. At the medical research I was doing, we researched rabbits and mice and so on. And how can you research cancer in a mice that doesn't have any cancer? Well, what you do is you give the mice cancer with what they call tobacco carcinogens. It's a long word. We never really said it. We just said NNK. So you take NNK and you spread it on the skin and they end up with skin cancer. You spread it on the breast and they end up with breast cancer. And so therefore we cause cancer to do research on cancer. And therefore we know there are many carcinogens which are very, very effective. NNK is very effective. It's cheap. It's, you just get it from tobacco smoke. So it's very easy to, it doesn't cost much. So we use NNK a lot.
So when your critics assert, and this is done frequently, there's no evidence for genetic entropy, there's no evidence for genetic degeneration, the accumulation of these mutation-related diseases, including certain types of cancers over time, is a line of evidence, a line of many reasons, I think, but it is one strong indicator.
Well, yeah, that's true. And you're denying reality when you say that's not a problem. I mean,
Right.
I can't imagine anybody in my field of medicine, if I said, well, mutations isn't a problem, we're getting less as time goes on. They would look at me like, are you okay? Feeling all right? they would recognize that you must be joking, because obviously you know that the mutational buildup is occurring in each generation. In fact, we can measure how many. We do a evaluation of the father, mutational evaluation, do an evaluation of the son, the grandson, and we can see clearly the increase in mutations has occurred from father to son to grandson.
That's some great answers, Dr. Bergman. To bring us back directly to the virus topic, from my understanding, we obviously have a lot of bacteria in and on us, billions, trillions of bacteria. But we, and a lot of people are shocked to hear this, we have more viruses in and on our bodies than we do bacteria and even cells, I think. And so would you argue then that a lot of those viruses are there to possibly regulate the amount of bacterial species that we have in and on our body? Are these regulating mechanisms, perhaps?
I think that well could be the case. Again, we don't know a lot about viruses, because I can collect money for doing cancer research, but collecting money to do virus research, people say, what do they do? What diseases do they cause? Well, we don't know. We're just trying to find out. Well, good luck with your curiosity, but I'm giving my money. My mother died of cancer, and I'm giving my money to the Cancer Foundation for good reason.
Yeah. Well, that's good, because typically your critics of biblical creation, they'll hear the word virus or bacteria and they'll think of something bad. When in fact, as you're demonstrating, the majority of viruses are good, and there's only a small percentage that are bad, but that's mainly due to mutations, damage, degeneration.
Right. And when you look at the virus size, even the bacterial phage was a pretty good sized virus. When you look at the size of the virus compared to the average cell, or the average bacteria cell, which is pretty small, you can see that viruses are tiny. You could probably fit, 500,000 in the average human cell. So they're very, very tiny. That's why they weren't discovered for a long time. They had glazed dishes, and they poured infection phlegm into them, and it trapped the bacteria, but the viruses went right through it. And then they took the filtrate, and then they put that on the skin or whatever, and then they found that caused disease. And so they realized that whatever was getting through the filtrate causing disease was really, really small, because it trapped all the bacteria. Bacteria couldn't get through the unglazed petri dish. And so therefore they realized it must be really, really, really small. And now we know, of course, it is really, really small, thanks to the development of electron microscopes and other techniques.
That's great. That's a great answer. This is great information, Dr. Bergman. So I'll get this next question up here. So here's the question. Can the evolutionary community adequately account or explain the origin of RNA viruses?
Well, we don't have much knowledge about the origin of any virus. So RNA viruses are a problem. I know they try to make claims relative to the origin of RNA viruses, but RNA viruses are retroviruses, AIDS is a retrovirus. And it's interesting that RNA has to have a retrotranscription. It's got to go from RNA to DNA back to RNA, mRNA or tRNA or whatever. So the RNA viruses kind of wonder why go through this extra step? Why would these be so different? Because if you don't have the reverse transcriptase, it can't survive. And so therefore it makes things more complex, and a lot of people wonder why. But to claim, when we compare DNA, of course, and RNA, we find a lot of strands are similar, and they make certain claims that came from viruses, and that's how we got it. But that's questionable. You can't really prove, I guess, didn't enter humans that way, unless you could get a corpse that was well preserved, that was 5,000 years old, that you could do analysis to see if they also had that RNA. My guess is you'd find out they did. And therefore they've been with us since the beginning. And many are really necessary for life. And so therefore that, to me, argues they were here since the beginning. They were added later on in evolution.
That's a great response. And so it makes me wonder and pose the question, since retroviruses like HIV, for example, as you mentioned, an exogenous retrovirus occurs from outside, since they require a host to replicate, isn't it to the evolutionist kind of a paradox? Because isn't the question what came first, therefore, the host or the retrovirus? But as it,
Very good point. Yeah, that's true. And you got to have both. The retrovirus, of course, since we know viruses, once they're alive and not alive, but once they're functional, and they can be dormant for thousands of years. But by and large, they have to at some time or another reproduce, otherwise the number will deteriorate. And so, yeah, that's a good point. They have to have a home somewhere.
right. So if what you're saying, and correct me if I'm wrong, a harmful virus, exogenous retrovirus like HIV, is due to mutations, damage, or crossing from one species to another where it can no longer be regulated adequately. So it would have essentially originated from a host at some point rather than evolved outside, right?
Right. And that's the assumption we make when we research these viruses.
Yeah.
In fact, we don't really make an assumption. We just say where they come from. They come from a lab in China, or they come from a bat in China.
Right. That's good. Yeah, I think the creationist has a ready explanation for the origin of these viruses. Okay, so the next question we have here is, and I know you've written on SIV as well. So the question is the change from SIV, from my understanding, SIV is essentially HIV in chimps or some primates?
Yeah, the simian
Okay.
IV, right.
So I've heard some evolutionists say that this change from SIV to HIV in humans is the addition of new information. It's evidence for forward evolution. What are your thoughts on that, Jerry?
Well, that's possible. When you say new information, it could be a mutation which converts one gene into something else, does something else, but that's pretty unlikely, but possible. But yeah, it's
if we could track back that specific exogenous retrovirus that's harmful to us, harmful to simians, but we could trace it back to an original host where it's actually beneficial to and not harmful, could that be the original created virus, or a virus that is pre-damage, pre-mutation?
Probably, yeah. And that's, if we go back far enough, I would expect every virus would be beneficial. Of course, most every virus is beneficial today. It's just they go from their host where they do fine and get into us, which they don't do fine. And so, but normally though, that should not happen. And normally our body should take care of the viruses. In fact, when you talking about going from the monkey or bat host to people, probably the person who picked that up first was not real healthy. And therefore it found a place it could thrive and then be spread throughout the area or the world, possibly, like in the case of HIV.
Good answer, Jerry. Next question comes in from Paul Anchor. Have they ever isolated a virus?
I don't know what you mean by isolate. I guess you probably could when they study them, but typically there's no need to really isolate them, I guess. I've never, usually you find them in such large numbers, like that picture I showed of a bacteria cell and the viruses surrounding it. So I guess you probably could, but you can't exactly take a pair of tweezers and separate them. You probably could. They're really small, and even with the electron microscope, you get a blur. And they have to do a lot of work inferring from this blur, although now we have better electron microscopes which give you more detail. The one we had at the medical college, I don't remember what it could magnify to, but it only goes so far, and you can get it larger, but it's just blurrier. So you don't necessarily want high power, you want high resolution. And my guess is, since I was at the medical school, we have electron microscopes which have better resolution, but you have a limit in resolution because of the light wave. So wave is the wave is so large, you can't get something smaller than the wavelength to get detail from it.
I appreciate it, Jerry. So next question is related, but I guess for a little context to the audience, we've been talking about exogenous retroviruses a lot of the time, like HIV, for example. But the question is, why do we share ERVs? So rather than exogenous, we have endogenous retroviruses occurring from within in similar genetic locations within other organisms, such as chimpanzees and primates. Dr.
Because
Bergman.
most viruses that I'm aware of, and again, there are millions out there, and I'm not aware of most of them, and no one's aware of most of them, but viruses tend to insert themselves in specific areas. This is why we can use them to replace genes, because they insert themselves. They don't randomly insert themselves typically. They insert themselves in very specific areas. And so, yeah, the fact that these endogenous retroviruses do so is, yeah, I would expect that. That's not unusual at all.
Now, would you argue that the endogenous retroviruses that we know to be functional in embryological development, for example, or in determining cell types, there's also many ERVs that are functional in the immune system. Would you argue that those are that way and are shared for design purposes, that they were created that way to serve those functions?
I would say, yeah, they're created that way to serve those functions. Pretty hard to prove that they came from elsewhere outside and got into our body and therefore found a place. Because you can't, unless you have to go back in time, and the time machines that I've seen don't work very well. So can't do that.
That's historical science we're dealing with.
Yeah.
And when these evolutionists say, well, we share all these ERVs with, let's say, the chimpanzees. Well, if we supposedly share them with the chimpanzees, that means all humans have them, all chimpanzees have them in that specific location. So it's fixed. But if there's a function for that fixed ERV, then why would we extrapolate that, like the evolutionists, to say, oh, well, that's the result of an ancient viral infection that eventually drifted to fixation? We can equally say that it's a created unit of DNA function. It's there in a similar location because it's created that way.
Yeah, I would say parsimony is the best rule, that we need to look at the most logical, the easiest explanation, and the easiest explanation is it's there for a reason. And it inserts itself in a certain part of the genome for a reason. It doesn't just willy-nilly. It would really cause havoc if it just willy-nilly inserted itself all over the place.
Right.
And that would be disastrous for the genome. And so a virus that did that, my guess is natural selection in this case would work, because they destroy the genome, destroy the host, and destroy themselves.
Right. And then the evolutionists would have to explain basically how that newly endogenized retrovirus managed to evolve the ability to be functional in placental development. I'd like to see them explain how that type of novelty can be gained. Okay, so my follow-up question is then, in some circumstances, Jerry, we find some unfixed ERVs. What I mean by that is, some humans within the human population, there's only a really subgroup, a small group of people that have a specific ERV. And so that might be, as you've said, since a lot of these viruses have preferential sites, it's not totally random, that could be explained through a true endogenized event.
Yeah, it could be. Not sure that's the easiest explanation, but could be a damaged virus, and the virus is damaged and therefore it doesn't insert itself where it's supposed to.
Yeah,
or there could be a similarity in the genome sequence, and it inserts itself in one area, which is not where it should be, but which is similar to the area it should insert itself in.
I appreciate the answer, Jerry. This is great. Comprehensive. And this is what I like about this topic. So in your book here, again, I've got it in front of me. People can pick this up on Amazon. That's where I got it. And so chapter 10 of your book, you discussed irreducible complexity and how it proves a designer. Can you speak to this a little bit, Jerry?
To me, that's an argument there's just evolutionists just cannot deal with. And therefore they use name calling. The old cliche is that if you have some facts, you present the facts. If you don't have facts, you attack the person. If you can't attack the person, you hammer on the table. That's what lawyers talk about, the three approaches in the courtroom. And it's obvious that so many things are irreducibly complex. And a good example is, of course, which Michael Behe noticed in the bacterial phase, you got to have all these parts. It cannot survive until it has all these parts. And we're not talking about gene mutations that can change these things. We're talking about a design system, which the genes are part of it, but the assembly is also part of it, which is not directly a result of the gene. So it's a result of the limits of, I guess, the natural world. And so just about everything is irreducibly complex. The eye obviously is. You have to have many parts, but for it to function. But one of the most basic ideas in biology, period, is irreducible complexity. When we study, for example, when you study anatomy and physiology, I taught A&P for quite a few years. When you study anatomy, they talk about the kidney. Okay, what are the parts of the kidney? So you go through all the parts of the kidney, and then they point out if this one's damaged, the glomerular system is damaged, it's not going to work. If this other system is damaged, it's not going to work. And so you're basically saying that for the kidney to function, you have to have all this long list of parts. You learn what the parts do. And if they don't work, the kidney dies, or in some cases, in fact, all cases that I can recall, the kidney simply doesn't work unless it has all the parts. And that's pretty much part of anatomy and physiology, because that's true with every organ. You've got to have all these parts before it works, period.
Amen. And the book of Genesis explains that everything was created very good. You know, these systems could not have evolved from inadequacy to precision to what we see today. And again, I think that goes back to what you were saying earlier, Jerry, in the law of parsimony. The most parsimonious explanations to a lot of what we see, and a lot of what you're talking about, Jerry, is creation, is design.
Right.
And so here's the next question, comes in from Brother Sid. He says, question for the guest. Any instances of bacterial evolution? So are there any instances of bacterial evolution increasing the functionality of its genome? I've only heard of it decreasing.
Right. As far as I'm aware, I've only heard of it decreasing. It's possible, though, that you'll get an increase in the functionality of the genome. That's certainly possible. It's just not likely. The problem you have to have for evolution to work, though, is you have to have a lot of increasing functionality. You can't have one or two rare cases here and there. You need massive numbers to evolve anything. And therefore, one or two examples possible, but one or two examples is not going to do it. Usually functionality is increased because something in the way is damaged. It's not
Right.
there anymore. It's reduced.
It's usually reductive.
And
And so would those instances then, Jerry, only really be localized increases in functionality? Much like if I wanted to temporarily get better gas mileage in my car, I could start removing weight, removing parts of the car that are there. But overall, that would be damaging. Yeah, it might be good in a localized manner. I'm temporarily getting better gas mileage, but overall, I'm destroying the car to do so.
That's a good point. That's true. And you have a lot of cars today you don't need, so you probably could reduce a lot. But in the long run, I'm not sure of that.
So since we're talking about bacteria as one of the topics, what are your thoughts on the long-term Lenski E. coli bacterial experiment? A lot of evolutionists argue, that this experiment has demonstrated forward evolution. There's been novel adaptive episodes observed. What are your thoughts on that, Jerry?
A lot of debates on that of what actually happened. But from what I'm gathering, what happened is that they put it in an environment which had, I think, some compound which normally the bacteria couldn't utilize as a nutrient, and this bacteria could utilize this as a nutrient. And I don't remember all the details, but much ado is made out of nothing in this case, that you're really not looking at any major evolution. You're looking at bacteria which evidently was able to utilize a nutrient, I think citric acid, which it couldn't utilize before. And so therefore it's not I would think he would really be discouraged with that, because after 20,000 years of evolution, basically he has minor things which he's claiming, but really you're not looking at much evolution at all.
Right. I agree. It's kind of funny that that's, one of their best examples of evolution. And in my personal study of it, in nearly every case of Lenski's experiment, genes were either deleted, broken, downregulated, or eliminated. So I think it goes back to this analogy of the car, removing all of this weight to temporarily get better gas mileage. Okay. In a local sense, that's okay. But in terms of total functionality, that's down. That's going downhill.
Right.
Okay. I think we're coming down to our last couple questions here. This one comes in from Echoing Erudite. Question: hypothetically, could bacteria or other pathogens play an important role in the original fruit on the tree of life that would greatly improve our lifespans?
First time I heard that before. I don't know. That's interesting. I kind of doubt it, but that's possible, I guess. I wonder. My feeling is, though, that Adam and Eve would have had the bacteria in their gut and in the outside of their skin. That they would have had that, would have been created with that, because that would be necessary to be healthy. And so, therefore, that was part of the creation. God created Adam and Eve, and Adam had gut bacteria. He had E. coli. He had all the, the bacteria necessary to keep him healthy. But it could have been on the apple. There was a pathogenic strain of E. coli, I guess, Escherichia coli, and that could have caused a problem. But never thought of that. But that'd make an interesting novel.
Yeah. Lots of interesting hypotheses and questions for us to explore. And that's why science is so fun. And there's always something to learn. And I like to say it's a great time to be a creationist. That's for sure. So do you think it's possible that just through over time after the fall, accidents in the packaging process within our genomes something could go wrong, and basically a pre-existing component of our genome could essentially go viral, and then that can spread to other. Essentially, a lot of what we see today is just originating in hosts, things that have gone bad.
Yeah, it could well be. Yeah, that's quite possible.
Okay. I'm just looking over these questions here. Oh, I missed this one. Jerry, you're a fan favorite. Every time you're here, people are never disappointed, put it that way.
Well,
which I should remind people, in the description box, right on our website, standingfortruthministries.com, I've added a new section called Guests on Standing for Truth. Click Dr. Jerry Bergman, and I've organized all your presentations on one page. And so you can start at the beginning and watch them all. Okay, so this question comes in from Matt. What was the biological purpose of the tree of life in the Garden of Eden?
I didn't know it had any biological purpose. I thought the tree of life was a symbol of tree of life was a symbol. And by taking of that, it's like mother says, you can't eat a cookie today. And so you deliberately disobey her. And then you claim, well, Mama, you didn't tell me, you didn't say I couldn't eat a cookie. Yes, I did. I told you no cookies today. You got to say that for dinner. So the fact is a behavioral allegation. It wasn't a dietary change. So although now I guess the claim is that the fruit was actually a pear. I'm not sure how they came up with that, but looking at Josephus and some other early Christian writers, they think it was a pear. But the apple's okay. It was grapes.
Yeah.
Grapes makes more sense.
Anybody with kids knows perfectly well the example you made on the cookies in the cookie jar before dinner. So this question comes in from Doki Doki Bible Club. And so his question is, what about those pesky parasites?
Okay. Well, we talked about a couple of them.
Yeah.
I'm not sure. Well, many parasites, though, are not parasites. They are not parasites. They live off of organisms, but don't necessarily cause problems. So you're thinking of parasites maybe that infect people and cause a problem. But of course, the leeches would be a parasite, actually. And the There are other examples, but it's interesting that in all my medical training and teaching, it never came up really much. Most parasites that are concerned are those that just get in the way. They cause discomfort, don't necessarily cause problems, although parasite usually referring to animals that need to live off another animal. They can't live by themselves. They don't make enough of the nutrients. They can't live alone. They have to live off somebody else because somebody else makes the nutrients they need to survive. So I guess technically we're parasites in some ways. So I guess it depends on how you define parasites. Maybe that's another chapter. I'll have to revise my book and talk about some parasites.
Always something to add, right?
Club should have mentioned some examples so I could be specific.
Could some of these microparasites be degenerated bacteria?
Could be, yeah. Evolutionists claim viruses are degenerated bacteria or degenerate organisms because they realize that viruses could never evolve. So therefore, they de-evolve from organisms that already have evolved at a higher level. So they're basically degeneration of, but that theory doesn't work very well, obviously, because what did some of these that we looked at, what did they degenerate from? What did the bacterial phase, what did that degenerate from? And then that's why right. Michael Behe looked at that and thought, there's no other explanation. Had to be created the way it is, right, for it to and I don't mean to use I guess live is okay, but as you know, they're not technically alive. They're gene machines.
Well, if they're just going to say everything's a degenerated something, well, it has to go back to a previous state that was not degenerated. And at that point, you're at the creation model. And I've even seen in their own literature, they're now proposing what's called the escapee hypothesis, which is very similar to what creationists argue. They're saying that a lot of these harmful retroviruses originated from healthy host cells. And so now even they're saying that a lot of these retroviruses evolved basically from within the hosts. And I think that supports a lot of what you've been saying tonight, Jerry, with your model.
Yeah, it does.
Okay, I think here's a question. Lisa, thank you for the question. She says, I was told that you need a virus in the body to even get cancer. Do you have any thoughts on this?
Well, cancer can be spread by viruses. The best example is the papillomavirus. What they do is they invade a woman who has cancer, and they then transfer her cancer cells into another woman. And therefore, the other woman gets cancer as a result of the cancer cells, which were transferred from one woman to another woman. They discovered this when they realized that a lot of women who got cervical cancer, especially, had husbands that worked as truck drivers. And so then they did some research and they realized that a lot of these truck drivers would have sexual relations with prostitutes at the truck stop, and they would pick up the papillomavirus, and then they would go home and have sex with their wife, and then they'd transfer it to their wife. And that was one of the main means they discovered indeed what happened there. Heard a lot of, I've never been a trucker, but you hear a lot of rumors about these gals that knock on the doors and wake you up at 3 o'clock in the morning and I want to sell sex, and you get pretty upset. I'm trying to sleep, lady. Could you please leave me alone? I got to drive 12 hours tomorrow. If I don't get sleep, I'm going to fall asleep at the wheel. So, lady, go bother somebody else.
Right. You know, these things are due to sin. It's due to the fall, mutations, environment. But overall, as you've pointed out throughout this show and in your book, again, I got to show it to the audience so they can pick up a copy. Excellent book. Viruses are good. You wouldn't want to live in a world without viruses. Bacteria are good. You wouldn't want to live in a world without bacteria. That's why after you take a round of antibiotics, what should you take afterwards? A round of probiotics and replenish that good bacteria, because bacteria is essentially good. And those antibiotics, they're killing your good bacteria and your bad bacteria essentially in order to wipe out the infection, Jerry?
Yeah, unfortunately, they kill both. They're not very discriminatory.
Yeah.
Some antibiotics are more discriminatory than others, but by and large, they're not awfully discriminatory and therefore kills the good as well as the bad. And doctors should, when they give you penicillin and other antibiotics, should tell you now you need to take some yogurt or eat something which will replace the bad bacteria, but they don't always do that.
Right.
And, I guess, you get busy and you have a lot of things in your mind and you don't always remind patients. But the pharmacist at least should always tell you when you take antibiotics. Now be sure and, have some yogurts too. Now, of course, many people are healthy enough that that's not absolutely necessary, but it's certainly good advice to just get some yogurt and reduce the likelihood of a problem in the future.
So if you have a viral infection rather than a bacterial infection, and you were to take a round of antibiotics, you're essentially doing more harm than good then in that case?
Yeah, I would say that's true. You can tell the difference in that bacterial infections typically are very localized. You have a certain area that's a problem, but viral infections affect the whole body. When you have pain from head to toe, chances are greater it's a viral infection. When you have very localized infections, a cut, for example, and it gets red and painful and so on, it's a bacterial infection. So generally, that is a way of differentiating. And you need to differentiate because antibiotics, as you inferred, generally don't help people who have viral infections. You take antiviral medication, which affects viruses.
That's very good. This presentation and show has been good for healthcare too, especially as we move into the winter where sickness is so common. So this comes in from Jessica. Should you wait a few hours after taking antibiotics to replenish with probiotics?
Probably doesn't hurt. I don't think because it takes time for the antibiotics to work. Maybe 5, 10, 15, 20 minutes, depends upon the antibiotic and depends on where it's a problem. So it probably wouldn't hurt to wait an hour or so that once it destroys the good bacteria, then you can replace it. I suppose if you take it right away, then when you take the probiotics, then it's going to kill the probiotic bacteria. So considering that, you probably should wait a few hours before you take the probiotic because you don't want the antibiotic to kill the bacteria in the probiotic. That'd be counterproductive. So I think they suggest waiting 4 hours.
Yeah, that's what I was
take
thinking.
The probiotic.
So going back to the parasite question, a specific parasite or class of parasites, you could say, tapeworms, roundworms. I think there's another one, hookworm. These are internal digestive parasites. What's the purpose of them? Were they once non-parasitic, or can you speak to that, Jerry?
I need to look up that. I remember when I taught this area, we talked about the parasites, but they're not a much concern in this country because a lot of these are a big problem in other countries.
Right.
And so, but that's a good point. I need to revise my book. I just came out with a new C.S. Lewis book. My first C.S. Lewis book was 150 pages, and people kept saying, "Oh, you forgot about this. You forgot about that. You forgot about what Lewis said here." And so I came out with a new C.S. Lewis book, which just came out a couple of weeks ago by a Canadian publisher, by the way. It came out and it's like 3 times as long. So it's much more detailed. So already as I interact with people more and more, people are saying, wait a minute, you forgot tapeworms, you forgot. So I need to revise this, although probably take a couple of years, but I need to revise it so I go into more detail about other parasites like tapeworms. Some of these can be pretty gory to think about.
Right. Unlimited work. If only we had unlimited time. This is your first C.S. Lewis book. So I've got this one. I think I've got your whole collection, Jerry. And you gave a presentation here maybe about a year ago on C.S. Lewis. So I'll have to pick up your second book. And
it's on Amazon as well. So
good.
And it's a different book. It's not just this is kind of the Reader's Digest version, the one you just showed the listeners. Whereas the next one is a lot more detailed and a lot more goes into a lot of details. And I have chapters on specific issues that C.S. Lewis brought up. But he was, as I mentioned before, one of the most effective anti-Darwinists of the last century.
Right.
Very, very effective. Even though he never talked about biology, he talked about the philosophical issues. And so that's neat. So my 2 books are the only 2 books ever been written about that aspect of C.S. Lewis.
Well, and a lot of your theistic evolutionists, they would argue he was a Darwinist, and you've demonstrated quite the contrary.
They do. We have a C.S. Lewis organization that meets in Indianapolis. And I asked if I could present my book and talk about it. And they said, nope, you can come to the meetings, but we don't want to hear your heresy, because he was a theistic evolutionist, period.
Yeah, they don't even want to question it. They don't even want to discuss it.
Maybe I'll send him my newest book and I'll say, hey, I'll come down. It's a 3-hour drive. I'll come down and I'll do a presentation. You can challenge me.
Right. Yeah, actually, that'd be fun. I'd show up for that.
Like to be challenged.
Right. And you're willing to put yourself out there to be challenged with live questions, live objections, challenges. And you are considering all the equations because you've now updated the book too to address anything else that has come in. So that's awesome. Well, C.S. Lewis is such a well-known, renowned theologian. And so the theistic evolutionists want him on their side. But you've proven that he wasn't. That's awesome. Okay, so I guess here's our last question for tonight. You touched on this in your presentation, so if there's anything more you want to speak to it, especially in the summer, what's the purpose of these mosquitoes? You touched on, if I remember correctly, they can be a food source, ecosystem balance. Is there anything else you wanted to speak to in light of mosquitoes?
Well, primarily pollination and dealing with certain problems like rats and so on, so they can be. And again, the males are totally useful. The females are I got to be careful when I say this. I had some quite laughter at audiences. Well, I didn't say anything. The audience member did, but the females are the problem.
The female mosquitoes.
Not the males. And then somebody chimed in and said, "Yeah, that's true of people as well."
Yeah,
he was only kidding. Of course, his wife
of course
gave a tap on the shoulder. So
you got to have a sense of humor, and laughter is the best medicine.
And
mosquitoes give us something to complain about in the summer, right? Oh, those mosquitoes! I got mosquito bites everywhere. So I think that basically covers everything. Jerry, again, I think you did a really neat job at concisely summarizing your book and really covering on all the major topics that you talk about in that book. It's an important book because your critics for years and years, and I'm sure you've seen it for years and years, that's one of their common criticism. Why, if there's a God, why would God create bacteria? Why would God create viruses, pathogens? And in my opinion, you have adequately answered that question tonight, Jerry. So what we'll do is I'm going to hand it to you. If you had any final words, final thoughts. We had a great audience. Everybody was edified. I was edified. I've been looking forward to this for a while. And so, as always, Jerry, I greatly appreciate the time that you give to us, as well as all the work you put in defending the faith, my brother. Any final words, final thoughts?
I do a lot of speaking. I'm booked up this month and next month. December, probably I'll be in Anaheim, California. March, I have an event, but I'm especially close by. I'm anxious to come out and visit churches and interact with people, especially small groups and young people. When I go and speak in churches, and I do a lot of speaking in churches, I finally speak to the crowd. I need to address the young people. And by and large, it just don't do that. It's a sea of gray, more and more. And they already agree with me, and they love my presentations, and they tell me so, and that's nice, but I need to reach the young people, which really I've been trying to focus on, because the young people need this information. The old people, the older people, it helps, but again, it's, they're already been Christians for 40, 50 years. There's no issue. So, but the young people, these are questions the young people have. And that will help, I know that will help answer some of these questions that young people have, that I did when I was younger. That's why I delve into all these things, because these are questions I had. I wonder, if God was God, why would he make pathogens? It doesn't make sense. So it's a question I had. And that's one reason I delve into this, to try to answer my own question.
It's a very fair question, and it deserves a response, and you've given an exceptional response. Jerry, it's nice to see that you've been out and about in churches giving presentations. I know, because I've seen several of your recent ones over the last, I would say, about 6 to 8 months that you've done at churches, and many of the topics you've spoken on. And so you're doing great work, Jerry. I appreciate it. Like you said, we got to reach the younger ones, because in schools, they're being taught evolution, anti-creation propaganda. And so I completely agree with you, Jerry. You keep up the good work. God bless you for doing this. To the audience, thank you so much for tuning in. I appreciate all the support, the feedback. It's great seeing everybody so engaged in this topic. And again, thank you for tuning in. Standing for Truth is out. God bless all.